The next speaker is Alex von Lühmann, and he's from from TU Berlin, and I'll let you say the name of your group.
Okay, well, thanks, Joe. Yeah, the name of my group is the Intelligent Biomedical Sensing Lab. Yeah, I'm excited to talk about a bit of, let's say, methods and hardware as well. So it's going to link quite strongly to what Joe started with. And basically, I'm going to talk about what we're doing in my lab and with collaborators to, again, to move DOT a bit more into the everyday world and thoughts on and methods that we develop to do multimodal processing and machine learning-based sensor fusion. Very briefly, let's see. No, okay, I'll use this one. Brief disclosure, I'm currently consulting for fNIRS. That is a company that is selling fNIRS imagers, but I'm not a shareholder, and my disclosure does not pertain to this presentation. Here at OHBM, there's a lot of fMRI, and what I think we already saw in the first presentation is that some of us in the optical imaging community definitely feel that there's a lot that we could connect to from the fMRI into the diffuse optical imaging communities between basically very well constrained to very naturalistic and potentially unconstrained environments. And so this is also what we're working towards with a particular focus on the challenges that arise from naturalistic imaging, which is mostly really variance in the data aside from the complexity of the actual brain analysis. So measurement artifacts, physiological confounders. The fact that we might not even have labels by design, so we might not have block or parametric experiments. And so the question is then methodologically what to do about that in terms of hardware, but then really also in terms of the analysis. So just to kind of recap, in other words, the motivation of the work that we're doing, we're focusing on diffuse optical tomography. The equivalence or the very close similarity to fMRI images was already brought up. There's a difference though, which is if we move DOT systems out of the lab and we move, then we have artifacts, we have confounders. There's a lot of cardiorespiratory and cardiovascular variance in the signal, and the question is how to address that robustly. And one of the answers that we're trying to push is similar to what Paul Thompson actually motivated yesterday. Multimodal machine learning is basically a great opportunity to, in this case, do sensor fusion and to explain away and remove and separate better brain activity from confounders in the signal, to ultimately get a good contrast of our brain activity under very challenging conditions. And this is also funded by the ERC. Since January, we are running a starting grant basically to try to achieve this goal. So brief recap of what we've seen in the first talk that I think is really good news for the community, which is that fNIRS instruments have not only become more wearable, as we saw in Rissa's talk. It's very easy now. I mean, not saying that it was easy, but to set up experiments in which people are naturally interacting because systems are wearable. But also these systems cover more and more of the whole head. And then if we increase density, we get to high density DOT. This was something very specific that some labs worked on, but now it becomes more and more available to the mainstream, which means that you don't really have to be an expert anymore to run these studies and to enter the same space. This means the image space that the fMRI community has been working in. So I think this is a great opportunity. This is why we're working on this, and this is why others are working on this. I want to show you two examples of what we've done to kind of move towards this, this goal of wearable DOT. You can see me here wearing another DOT system. We saw Joe's system. This one here combines a 3D printable cap that basically allows us to pretty flexibly design our holders. On a head model, can be individual or an atlas, print the cap, and then, more importantly, use a system, which is in this case the NIRS, which is a custom-built system that puts, you could say, conventional optodes, but in a very high-density configuration into this cap. What you end up to, in this case, in most of our studies, we have around 200 optodes in total. Supported are about 250. What you then get is, well, one of these typical images. So on the left side, you can see the channel space. And then if you do image reconstruction, you get brain activation in image space on the cortex. Briefly, pointer is that what we did, and this is very heavily driven by Boston University, so our collaborators, is open source. So all of this is available on the openfnirs.org website /hardware. So you can basically download all these files and build your own system, at least in terms of files, and also now a manual for documentation. All of this is there. So this is, of course, an open source project, and there's other open source projects, but as I mentioned before, commercially there's now also systems, so it's not really a reason to not do DOT studies anymore. So how we use this system in my lab, what you see here now is just a video of that same system. In an experiment that I'm going to briefly pitch to you, we're currently still analyzing. It is run by my PhD student Bilal Siddiq. The idea here is that we do a dynamic functional connectivity analysis during natural interaction, where, this is me again, we're wearing the DOT system, we're wearing an eye tracker and additional physiology. I'm going to show you in the next slide what kind of physiology we are acquiring. We do photogrammetry, similar to what Rissa showed, so basically to make sure we always know exactly which signal comes from really the individual brain area. And what we now can do, and everybody can do because of the existence of these powerful open machine learning models, computer vision and text-to-speech transcription, we can do scene analysis in this experiment from the eye tracking and scene camera. So what we have in this experiment is certain blocks in which, in a self-paced way, the participant plays Tetris on a tablet, stares at the wall in a resting condition, or has a conversation with the experimenter. And all of these conditions can be automatically detected, but we are, of course, now interested in looking at the actual brain dynamics. This is the part of the let's say, hardware. So I'm going to show you a bit more of experimental setup itself that is enabled by this wearable hardware. Here is an experiment in which we look at motor decoding. The idea is to do single finger decoding using wearable DOT, EEG, and more. So here you can see how the setup looks like right now. It still takes a bit. It's not super convenient, but it allows free and naturalistic motion. What you see is that we have co-localized DOT, EEG, so high-density optodes and EEG in one spot. This has been recently published. But we also add a capnometer, so for end-tidal CO2, non-invasive blood pressure, respiration, GSR, so galvanic skin response, PPG, and we have our custom, you could say, joystick with which we we log motor activity, but we also create feedback through a little vibrotractile stimulus during button press. So this is led by my postdoc Thomas Codina, and again, you're not seeing any analysis results yet because this is work in progress. What I wanted to show here in this first part is really what the hardware that is now out there, that we are developing, that others are developing, enable us to do to try to push forward towards more naturalistic and certainly challenging data analysis. And then now really the data analysis part. What I just showed in terms of using all of this additional physiology is, again, is I think exemplary. We're not the only people that are doing this. There's a general trend towards multimodal integration to explain systemic physiology in the fNIRS signal. And more people agree that, similar to what has already kind of pretty much been established in the fMRI community, that modeling physiological signals into the fNIRS or DOT decomposition is helping and should be happening much more. But there's not yet really a full set of established tools for this. And also, if you now want to do machine learning and use data-driven approaches for sensor fusion, as I promised in the very beginning, there's also not yet really a set of established tools. So it's kind of bad news, but it's also, again, a big opportunity because there's a lot of things that can happen, that can be done. In my lab, we're particularly interested in trying to get towards single trial decoding. So the idea is really like if you improve contrast, we have better statistical power, but we can also do decoding, which is something else we're going to hear about in the last talk of this session. But so with the, let's say, motivation and the opportunity that this is very promising, but there's a lack of tools, we're trying to fill this gap with the work in my lab, and so we're trying to really push multimodal machine learning side to explain physiology, explain variance via data-driven approaches. To combine this with state-of-the-art imagery construction, which is really has been established over decades by people like Joe and others, and combine this with really what is now possible, as you saw in the two experiments that I showed and that we saw before, using wearable multimodal DOT. And so we didn't want to reinvent the wheel. Sedalion is the name of a toolbox I'm now pitching. The idea is Sedalion in Greek mythology is a dwarf standing on a shoulder of a giant. The giants here are the Python ecosystem, other community toolboxes like Neurodot, other neuroimaging toolboxes like MNE. And the idea is of this toolbox that we're basically bundling, you could say, established and also novel signal processing methods from fNIRS and DOT. We have a photogrammetry feature. We combine this with photon simulation support and imagery construction to get into image space, to get into this space that fMRI people are used to, and we are more and more getting used to in my lab. And then we are already in this environment in which the Python ecosystem offers, as we all know, a lot, amongst others in machine learning, so scikit-learn, PyTorch, and so on, to do sensor fusion, latent component analysis, and so on. The idea is to do this following standards. I mean, bits, everybody will know. fNIRS is also, the fNIRS people, I think, are slowly but surely also establishing the fNIRS bits standard. So we are all in the same kind of system. And using SNRF as a file standard is supposed to allow us to switch between toolboxes and pipelines rather easily to kind of always take the best of all worlds that are there. Just before this conference, we've released the kind of next bigger release. And so if you're interested, you're welcome to check this out. You can run this toolbox also on the cloud on Google Colab to just see how it works for you. A typical pipeline similar to what Joe has shown that you can do is if you have a segmented MRI, either an individual or a head model, an Atlas head model, you get your head model in the toolbox. You can co-register with photogrammetry the subject's optodes. You can do forward modeling, so photon simulation, and the actual image reconstruction. This is here now showing, this is my brain, so you see on the left hemisphere activation over the motor area during my right-hand finger tapping. And as I mentioned before, now we are in this space in which we can very easily do what Paul Thompson mentioned yesterday. We can apply machine learning. We're already in Python. We have all of these packages. And I'm going to briefly show a bit of more results on the next slide. So I'm not talking too much about this, but you see here how classification performance of single trial decoding of motor tasks improves with the number of channels on the cortex. So this is what I'm going to briefly tell you now. So first of all, to say we have not done any, you could say, state-of-the-art model selection or channel pruning, artifact rejection, all the things that one usually does to optimize decoding performance. The idea here is to use on a rather small example data set to compare really a few very simple parameters. The first one is the number of channels. So if you have a sparse fNIRS system and you go into higher density fNIRS systems, what you can see here is that the classification performance, single trial, left versus right finger tapping decoding performance continuously increases with higher density. This is still using channels, not image or voxel space. And that if you correct for systemic physiology, that's the orange line, we get additional performance boosts. But now the really interesting thing, so this is kind of one thing, that's higher channel density helps. But if we have high channel density, then we can do what we've been talking about here, which is diffuse optical tomography. We can go into the image space that, fMRI people are used to. And so what you see here is just a comparison, same data, of classification accuracy between channel space, so using what we showed on the left, and image space, parcels in this case, on the y-axis. The gist is basically that you get quite a significant improvement of 5 to 6% with quite a large standard deviation for this decoding, which is just kind of highlighting once more what most people in the DOT domain already know, which is that it helps us getting better spatial-temporal contrast. And on my last slide, because I talked about multimodal, I want to briefly highlight currently ongoing work. So this is by Tomas Kodina. I mentioned him before. He's doing this DOT EEG decoding study. What he has established is a pipeline that helps us to use real, concurrently recorded DOT EEG data and augment it with synthetic ground truth, so with co-modulating sources on the cortex, so a dipole and a hemodynamic response that are realistically modulated and augmented onto the resting state data, which basically gives us something like a hybrid between perfect toy data on which every machine learning method somehow performs, and realistic data, which is actually then the challenging case that we're really interested in. He implemented basically the whole family of canonical correlation analysis methods, constrained, unconstrained, regularized versions that are all part of Sedalion now, and this allows us to really evaluate how well these methods are working on semi-realistic and then realistic experimental data, and how to do data fusion between fNIRS and EEG, or DOT and EEG best. The other thing that I'm very excited about, I think this is now not really time synchronized, unfortunately, is learning from a shared space. So DOT and fMRI, as we have seen now, share quite a lot of information. So we have a postdoc and a PhD student that are trying to apply deep learning to basically take what the fMRI community has given us in terms of data, but also in terms of insight, and apply it to the DOT domain, which really is a quite straightforward transfer. And with this, I want to close, just to emphasize, I think this is really where an opportunity is to really kind of link both worlds and to move from a very constrained, very well controlled environment into naturalistic. So it's not really competing, but complementary. With that, I would like to thank particularly everybody involved, my team, and also our collaborators at Boston University. And I pass on the torch.